with a PhD in molecular genetics. His main focus of research was epigenetics and evolution. And while he was here, he also got an MBA from the Terry School of Business at the same time. And now he's the COO of Vector Builder, which is a gene delivery technology company. And he'll be telling us all about that today. So everyone, please welcome Dr. Mussar, and we'll get started.
Thank you all. Yeah, Dr. Mussar still sounds weird. I believe Mary Bedell has retired this year. But I used to call her Dr. Bedell, and I remember one time in the elevator, she yelled at me and says, "Call me Mary." Never call me Dr. Bedell ever again. So thank you all for having me. It is really truly an honor to be back 10 years since getting my PhD, in May of 2013. And I hope this talk is really, really beneficial for not only some of the people who maybe remember me and to see where I am today, but also the second half of the talk is really geared to a lot of the grad students and postdocs and research scientists who maybe are hitting a point where they want to, reassess and think about what options they have in life. So with that being said, we'll get started. And really, I want to talk about scientific advancements first. And there's usually two general approaches how we could address scientific advancements. And one is, more where discoveries may expand upon or improve on existing technologies or an idea. And this is that linear approach. This is what we're very used to, building on, our predecessors. And it's very important. But then there's also this side on the right where you may forge new paths and disrupt the existing paradigms and branch out and really try to redefine fields and new ways of thinking about it. And it's really important to note that both types of scientific advancement are necessary to push science to the next step. Scientific advancements can't solely be linear. We need to, always be thinking outside the box. And if you ask some of the professors who are still there from my days, I think they would agree that I was never a go with the flow type of person. I'm a contrarian by nature. And now there's this new term called disruptor. And there's a nice little article about me from Authority Magazine on how I disrupt the molecular biology field of gene delivery, which we'll get into later. So we're constantly asking ourselves, what's going to be that next big step? What's going to be that next breakthrough? What's going to rock our worlds next, right? And, many breakthroughs are really proving to be elaborations or improvements on what came before it. Of course, there's exceptions to this rule, and, many are driven by, biotech resources. For example, during the pandemic in 2020, they really, opened the door and gave us the opportunity to really show the world what we're capable of. And, with, VC funding, which, some of you might not know is very down this year. VCs will dictate the ability to think outside the box. But, studies are showing that truly novel disruptive breakthroughs that drive research and understanding in these new directions are becoming less common. And there was a publication this year in Nature that really kind of tried to quantify this. And it's not true for the life sciences, but it's true for all of the sciences, social sciences, technology, et cetera. And they've seen over the last 60, 70 years that these disruptive papers are becoming less of a thing as we worry about funding and so forth. And, Connie could share this paper or I can later, but they really tried to quantify disruption by looking at the verbiage used in publications and whether the verbs were more about, building on previous advancements or trying to facilitate new breakthroughs. And they saw that, the verbs used in publications kind of have driven away from that disruptive, nature and more into that follow the leader linear type of pathway. So that brings me to what I want to talk about in the first part of this talk. And that is something that is really near and dear to my heart. And that's gene delivery. I say that because when I was starting my PhD in Rich Meager's lab, now 15 years ago, I was put to work straight away by, cloning, multiple constructs. By the time I left UGA, 4.5, 5 years later, I like to think I was considered one of the top people in the department to go to with their cloning and gene delivery questions. I say this because people came to me with their cloning and gene delivery questions. But I wouldn't be there without the help of, all of UGA's faculty, especially Rich and Elizabeth McKinney, who was, like my lab mom, and she was our lab manager. Kanasami was our research scientist, and I learned so much from him. And I do want to single out Daewon. He was a postdoc in Jan Westphaling's lab, and he was my sanity check. Him and I would be working nights and weekends. And when I was so frustrated with running a reaction, he would, help calm me down, help me troubleshoot. And he actually, even, ran some of the reactions for me to make sure I wasn't crazy. So, big thanks to everybody, at UGA for helping foster my love for gene delivery. So I want to ask a question to the people, listening or in the room. And by a show of hands, how many of you are introducing a gene or a gene fragment or some sort of genetic element into an organism right now? Can I get a show of hands? 1, 2. You're supposed to be doing it. So it's very easy to see how they're like really shoehorned into specific, directions, considering the complexity of all of your fields. But gene delivery is wide and it's very varied when considering how to get your genetic material into your target cells. And there's a huge number of considerations to, factor in. What application are you using? What's your question? Do you want to knock down something? Do you want to knock out something? Do you want to overexpress? Are you testing enhancers and promoters? There's a wide variety of applications you could be asking. And then, what is your model organism? When I was at UGA, I believe we had 55 model organisms in 60 labs. I don't know if that's changed at all, but, there's constant variation of the organism you're studying. You're studying mice, lizards, snakes, fireflies, beetles. I was studying Arabidopsis actually, chickpeas. There's so many different things that you could study, and this, factors into the type of vector you're going to want to build. Another question to ask yourself is, do you want to use a viral or a non-viral delivery system? And I'll go on to elaborate on this, moving forward. Do you want to control expression? Do you want it to be stably integrated into your genome? Do you want it to be transiently expressed for a short term? Of that expression, do you want it to be ubiquitous throughout all tissue cells, tissue types and cell types? Or do you want it to be more cell specific? And, this goes on, and much of the answers, that you researchers come up with to these questions are usually depend on the experience in the labs that you are trained. We're creatures of habit. I know that, even when I was graduating, I was in the dark room, making my Westerns. It took me 8-9 hours, the old fashioned way, even when they had those 30-minute Western kits. So we do, tend to pursue what we're trained in and what our PIs do. But I really implore you to realize what's out there and think outside the box. Gaining understanding of a system is often a long and tedious process. And, to understand all the complexities of cloning and introduce your genes of interest and producing your final product and knowing, the transduction, transfection process, anticipating outcomes and potential hurdles can feel like a really risky investment. And you say, I just want to get out of here. I want to graduate. I just want to publish, and just get something out. So the tendency is to ensure that the research question matches the known approach. But this is very limiting. And it makes it difficult to push through to that next big discovery and really turn the field upside down. And that's what, I think, me personally, that's what we should be striving for, because we're at the time where we can make those big discoveries to make those big changes with the information we know. So, here in this slide is just showing a couple pictures of the different type of systems. We're all familiar, I'm assuming, with building a regular plasmid, and then doing, transfection, electroporating our cells, and then hoping and crossing our fingers for the best that it goes into, where we want it to go in. But now with technologies we have, we could turn to a more viral and more directed approach. So on the bottom here, you're looking at just a lentiviral backbone. that you would, insert your promoter and ORF and any kind of markers you'd want. But then with viral delivery, you're going to have to, incorporate packaging plasmids, transfer plasmids, envelope plasmids, etc. And then to build your package and activate your live viral particles to then go and infect your cells of interest. So that's what I want to elaborate more on, because this is where, the gene delivery field is really going, is harnessing the powers that viruses have to, really specifically achieve all of our gene delivery questions and what we want to do. And you're very lucky because there's resources that exist to help in both design and the synthesis or ordering of your vectors that already contain everything you need in a given system. And this is a not so shameful plug of the main part of our website here at Vector Builder, where we have over 150 different gene delivery vehicles that you could choose from to answer your specific question. And the beauty is, I understand you're not all molecular biologists, so you're not going to know all the ins and outs of all these systems. But that's the beauty of a company like Vector Builder. You come to us, you just ask us a question. I need to express this in these cells in this organism. We could help build and design that vector for you, which you can then take and make yourself, or you could, add to your shopping cart and order from us. So again, we're a free resource. We're trying to really help, empower all of you to not be afraid of molecular biology and what it really holds, and to instead harness all these new technologies wherever you are in this world, and really, be able to make those next big discoveries. So let me talk about a couple of systems here, just really quick to give you a flavor of what we could offer at Vector Builder and what your options are just in the gene delivery space. So first, we'll talk about using lentivirus to do, let's say, an shRNA, knockdown. I assume a lot of you are familiar with RNAi or RNA interference, maybe like in worm. I'm assuming that the curriculum still contains this from my days. So right here, we're looking at some figures that are showing, lentiviral vectors containing shRNAs. And, just like with many of the questions, there's options. Do you want to use a U6 shRNA vector, which is going to target only a single, transcript and have, relatively higher efficiency? Do you want to be a little bit creative, and maybe you're working on an evolutionary thing like I was, and you want to target multiple, multiple transcripts in a gene family to see the effect it has on, the cell structure, et cetera? Then you could use something like Mir30, which allows you also to incorporate, your promoter of interest and target multiple, different transcripts in one go. Now, as you could imagine, Mir30 might not have the efficiency as U6 would, who's only targeting a single transcript. And you have to really incorporate drug selection to fully realize your knockdown. But, it's a fairly simple process. You build the vector either yourself or with us at Vector Builder. You're going to package your virus. You're going to do your transduction, the hex cells or whatever cells you're studying. And then you're going to do your flow cytometry and your drug selection to really, see how much that the shRNA knocked down your gene. But there's other options we could go with as well. We could do lentivirus to do knockouts, and we could harness the power of CRISPR, which I know all of you know. So you could actually contain a single vector that uses all the CRISPR components to lead to a knockout. You could package your lentivirus with all these components, transduce it into EGFP expressing cells in vitro, and then measure your knockout efficiency via flow cytometry, which you see in Figure A. However, as going to be the theme of this talk, there are options you have. You always have options. And, do you want to go with a single vector or a multiple vector? What kind of CRISPR forms do you do? Do you want to use CRISPR-I, CRISPR-A, CRISPR-T, CRISPR-XYZ, whatever the new CRISPR is coming up? And this would be a great question if Michael Turns is listening. So here at the bottom, we're just showing again, fluorescence analysis in B. Of your knockdown. In C, you're looking at PCR amplification of your target region. D is going to be your flow cytometry. And you're basically looking here now at, quantifying from your untreated control, your non-targeting control, or what we call a scramble, and then your experimental gRNA treated cells. So again, using lentivirus, previously to knock down with shRNAs or knock out with CRISPR. So viruses really have so much capabilities to achieve any type of gene delivery you want. I discussed briefly lentivirus, which, what advantage it has is that it stably integrates into the genome. It has genome integration. Other viruses of, in this sort are MMLV and MSCV retroviruses, which are historically used, for, the generation of different types of stem cells, and so on. But I want to turn our attention now to the other side, and that's using viruses in these kind of vehicles that could, deliver your genetic material, but without integrating into the genome. So just a transient expression. These are the adenovirus or adeno-associated virus, abbreviated as AAV, which I'm sure many of you have read about in recent publications. AAV is actually what is our bread and butter at Vector Builder and is by far our most popular gene delivery vehicle. Now, but the disadvantage of working with AAV is its really small cargo capacity. It can only really, here on the slide, you see it says 4.2 kb. I could tell you we could make it work up to 5, maybe even 6. But when you get over 4, it gets very difficult to start packaging it. So to kind of circumnavigate this drawback of AAVs, we have basically optimized and worked with other viruses that could basically not integrate into the genome. And this is some examples are VSV, vaccinia virus, and herpes simplex virus, HSV. And a few that are not listed on here that we have now since optimized and launched are Sendai virus and rabies virus. So I know, you think rabies virus, some of the older people in the audience might think Cujo and Stephen King or something, a little bit more getting bit by a raccoon. But again, all of these, we're stripping apart we're all of the infective and bad parts of these viruses and just using them as gene delivery vehicles. They're just shuttles. And it's amazing what these viruses can do. So why not harness their power? So I want to talk a little bit more about AAVs because they are so popular, especially as we push into gene therapy. And oh God, the sun is right in my eyes right now as it says. I apologize. AAV, the beauty of this is going to be specificity. So when working with AAVs, they have these, capsid proteins on their cell surface that are going to bind to different receptors of the target cells. And, basically, altogether, the sum of those capsid proteins are going to be called a serotype. And there's many different known serotypes of AAVs already, that already exist that target all different types of specific tissues. And there's also AAVs that are more, that are targeting multiple types of tissues. For example, here in the bottom right, this is AAV9. So AAV9 is notoriously known to target many different types of tissues. If you want more ubiquitous expression, you can see it's the hippocampus, the retina, the liver, the pancreas, skeletal muscle. So this is a serotype. If you don't really know what you want to target, this will target all of them, and you could pull out the cell types and imagine them. But there's also serotypes that will only target maybe neuronal cells, only target liver, only target skeletal muscle, and we're constantly trying to develop new types of serotypes. So this is a process called AAV capsid evolution. And it is actually one of our, we're one of the few companies that are able to achieve this. And was on the phone earlier with somebody from Germany who's also going to pursue one of these projects. So in a capsid evolution is basically you're trying to develop a new kind of code of capsid proteins on the outside of this virus. So you're going to develop a capsid library of all these different types of proteins, and then you're going to go look at different combinations of them to try to develop a new serotype that's going to target your cells specifically and nothing else. And it is really amazing what you could do with this type of technology as we continue to go. And we're actually at Vector Builder really, really pride ourselves on creating an AAV superbank, which is basically like we want to continue to do this, even if we're not paid for, to develop more and more serotypes to create more specificity when using these viral vehicles. So, the future is very sound when it comes to, what we could do with specifically targeting your cells of interest only as opposed to, not knowing. And as you can imagine, when you're talking about gene therapy and bringing a viral drug to market, the more specific you are, the better. It'll get you through the trials quicker. Trust me. So up until now, I'm, really talking about a lot of examples, and I understand I'm talking fast. It is, a little bit later for me. I'm kind of winding my day down, but we've been talking a lot about reverse genetics approaches, like functional studies, clinical applications, but it is important to note that there's forward genetics tools as well. And this is more like, creating libraries to try to identify, targets, drug targets, genes of interest in your pathway, basically areas for you then to go and maybe back to a reverse genetics approach to study. So it really is an invaluable tool, but it is very complex, especially when you consider the variety of options available. Do you want to make a CRISPR library? Do you want to make an shRNA library? Do you want to use one vector or two vector system? Do you use dual or single guide RNAs? And I know the answer to that question after negotiating our recent CRISPR license. Use single RNAs. Dual guide RNA libraries? No. Just to let you know. So, and the number of gRNAs and sRNAs, how many do you want per gene? Do you want to have multiple guide RNAs targeting each gene to, really be more specific and trust the data more? Do you want to use singles? The more complex you go, the more, the harder it is going to be to create the library, or the more costly it is for a company to create the library. But the options are out there. So whether you're using reverse or forward, what kind of question you're going to ask. Maybe you're just starting your PhD and they ask you to study a pathway, and the best thing to do is a forward approach to go find a few candidates for you then to go take and really dissect and study. Furthermore on this is, developing stable cell lines. And, you could do this for more efficient screening and studying, gene function and modeling diseases and really trying to decipher mechanisms. Again, you could use, a CRISPR approach. We actually have this modified proprietary CRISPR directed recombination efficiency. And a higher percentage of successful point mutations are usually you're going to, basically, depending on if you want, the theme is there's many different options to go to for your specific research question. But, we here at Vector Builder are here to answer those questions and direct you into the best model possible, given our, extensive experience with researchers literally in 90 different countries throughout the world. So what you're looking at here is an example of, if we're going to do a point mutation knock-in. So, just for all of you who are going to try to make stable cell lines yourself, if your PAI asks you to make a knockout stable cell line, tell them, yes, I could do it. If they ask you to make a knock-in, don't agree so fastly. Knock-ins are exponentially harder than knockouts, for reasons you could probably infer. But here on the figure, you're just going to be looking at, the generation of a knock-in stable cell line, where you're gonna create your donor vectors and your gRNA vectors, electroplate them, generate a pool of either pooled cells or monoclonal cells, and then you're going to do your testing. And testing for a point mutation knock-on is relatively easy. You're going to go look and you're going to do Sanger sequencing and basically go look for those point mutations. And when you introduce point mutations, you're obviously going to have, synonymous and non-synonymous. And what you're seeing here in the red box in figure A down in the bottom right is going to be representative of a non-synonymous mutation, whereas the green square is a synonymous mutation and something that you might be looking for. So to kind of close out this gene delivery part of my talk and get into the fun stuff, don't worry, it's going to get more fun. But I want to talk about something that's like really on the tip of many, many people's tongues, and that's mRNA delivery. So using mRNA as your vehicle. This is, when I say this, I'm referring to things like lipid nanoparticles, or LNPs, for those of you who are reading up on them. And, delivering your genetic information via mRNA gene delivery seems actually easier, right? There's less, you're cutting out a step. It seems like it'd be actually an easier process. But for any of you who've worked with RNA before, it's a fickle molecule. it is really tough to work with. There are so many things that could go wrong. And, you're down in Georgia, just make sure if you're working on mRNA, that the air conditioning's working, because temperature will just kill everything that you're doing. So, the good thing is, in the last few years, there's been vast improvements and optimizations to mRNA gene delivery by basically trying to optimize every part of the molecule. Each of these optimizations could actually take a researcher many years to perfect. It could definitely cause a graduate student to gain, many gray hairs, or in my case, lose their hair, which I did lose my hair during my PhD, but I don't blame the PhD. I think that's more genetic. It can make people cry in the lab trying to do these optimizations. And when I talk about this, I'm talking about optimizing, the UTRs, going back to stability. It's trying to change the poly A tail lengths. Obviously, if you really need it to be stable, trying to extend it, and, trying to synthesize a string of straight A's is just really, really difficult. And then optimizing things like the Kozak sequence. And then the final thing, which most of you are all, I assume, familiar with, is ORF codon optimization. And this is taking your open reading frame and really using these algorithms that are out there to optimize it to be the most efficient it can be. Of looking at what we did is basically we're showing you, we did gene optimization on a luciferase gene, and we created this new optimized, what we called high express FLuc or FLuc. And this is basically a highly optimized luciferase that you could see both in vitro and in vivo is expressed higher and far brighter than its wild type counterpart. So I could tell you also that we just broke ground, and we are trying to finish our facilities to create our mRNA gene delivery center to allow actually to do gene therapy on the mRNA front, which obviously, since Moderna came out with their vaccine, is a very, very popular pathway to follow. Although I do warn all of you, do not go looking for a job at Moderna, because they're all applying to us right now, and everybody is leaving there. So anybody investing, no Moderna. They hit their peak. So you can see, talking about gene delivery, and it really is just something that is applicable to all of your research, and it's just a fascinating field of what we could do. And I've always loved just trying to, understand cloning. And I put this flyer together, when the company was first starting, to try to, really, pinpoint all these major discoveries that we sometimes take for granted. Isolating DNA ligase, isolating the first restriction enzymes, to the invention of Sanger sequencing, to what I think one of the biggest inventions of all time is Carey Mullis in the early 80s inventing, PCR and amplifying DNA. Onto the turn of the millennium, where we had, we finally be able to synthesize, and, it set the groundwork for synthetic biology as we know it today. And then as we, continue to come up with new kind of cloning and DNA assembly methods like Gibson assembly, Golden Gate cloning, and so forth. And I really hope that, our revolutionary design and ordering platform, Vector Builder, could hopefully one day be, on this roadmap of major discoveries in cloning. Because let's face it, cloning is going to be obsolete in 10 years. What was previously thought of an active passage or initiation ritual for any molecular researching entering their PhD will soon be gone. I mean, I was initiated this way, and I'm better for it. But I know that 10 years from now, people are never going to have to clone themselves. The technology, the cost, and the effectiveness, and the difficulty to publish is not going to be based on this. I was asked to publish a paper just on assembling a certain vector back when I was at UGA. That you can't publish building a vector anymore. Times are changing. So, we say at Vector Builder, let us build these complex reagents for you while you worry about the downstream and more important questions you're going to ask that are going to change the world. So, now that we've done that, which, that took longer than I thought. I want to get into, what you could do with a PhD from the University of Georgia, and give you a little story about, me and my journey, and what some of you grad students and postdocs could look for, and what are your options? So I started actually in the University of Illinois in Urbana-Champaign in organic chemistry research. I was doing MRI on the big machine, NMR on the big machines. I was doing metathesis reactions. I was actually translating the old Deutsche Berichte from the early 1900s to creating new reactions. So, that was my initial passion before, shifting to a structural biology lab. And I was really lucky enough to be mentored by the late and great Dr. Carl Woese. So could I hear someone, since I can't see you all, can I hear a yes from anybody who's heard of Dr. Carl Woese before?
There's some yeses.
There's some yeses. Okay, there's some yeses. I really appreciate that. He was actually going to be, I tried to invite him as my student speaker when I was there, but unfortunately he was too sick to travel at that time. So those of you who don't know, Carl Woese is the founder of Archaea, the third kingdom of life, and the inventor of ribosomal RNA sequencing. And he really changed it, because at the time, this was 2005 or so. I took the MCATs on my 21st birthday. I was going to go go into medical school, and he told me to really rethink that decision. He said, you're going to go to medical school, you're going to learn the same thing, countless other people know. But he saw something in me, and he knew that I was too curious to just follow normal, medical texts. And he pushed me to really think about what the next 20, 30 years, what are going to be the big things. And back 20 years ago, I said, it's going to be computers and it's going to be genetics. So as you can imagine, there's a field that is now very popular, but wasn't really popular back then, called bioinformatics. At the time, there was only 2 grad programs for bioinformatics, and I think it was in WashU in Seattle and George Washington University in DC. So I went to DC, got a master's in bioinformatics, and worked on developing algorithms to discover novel non-coding RNAs. I made a bunch of in silico predictions, and then my committee asked me, are you going to validate these predictions? I told them that's what a PhD is for. So I did realize this, and I realized that having really good knowledge on programming and coding and developing, in silico, kind of predictions is great. But if you can't validate them, what's the point? So my molecular skills weren't that good. So I decided to go, after interviewing many places, to the University of Georgia, and I'm very happy I decided there, over other schools. I love to play golf. I think Bubba Watson was playing at the time. I figured you had a great golf course there. I could play almost all year round. I loved the faculty there. I was, really, attracted to the evolutionary stance. I wanted to kind of bridge the gap between the evolutionary side and the molecular side. So, I was really happy to make that decision. And while at UGA, as alluded to, I was actually the first person in UGA's history to get a PhD and MBA simultaneously. They actually weren't going to let me into the business school at first because they didn't know how to build me. And I'm really happy that I did it, because I paved the way for all of you to follow in my footsteps. And, someone in the faculty could correct me if I'm wrong, but I believe John O'Neill actually followed me a few years after I graduated and also did a PhD and MBA simultaneously. So I don't keep in contact with him, but I'm really happy to see that, I opened the door to thinking about other things. And maybe you have that entrepreneurial mindset. So, I could tell you your PIs might not be thrilled. And I know that I was on the outs with many of them, but I never look back, and I was really proud of that. So after graduating from UGA, I went to a company called GenScript. Now, I don't want to talk about them too much because they are our biggest competitor right now. I worked there for a year, and I said to myself, I could do this better. They are not for the researchers. They are a money grab. They will just want to take your money, and they're all about, corporate environment, profits, profit margin, the bottom line, blah, 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 blah. I said, this isn't me. I care too much about science and making it accessible to people all over the world, whether you have funding or not. And I guess, I'm just I believe in this utopian society where we all treat each other the right way. So, after working there for a year, I just said, no, this is not the right thing. So I left. And I actually went on to become an entrepreneur. I started, I think, 2 companies that failed. And that's an important thing to note for all of you. You will fail. I failed, and I'm better for it. You know, after starting a couple startups that didn't pan out, I actually was like, okay, maybe I need a little more experience, you know, working for smaller companies and building them up. And I came across in 2015 a Howard Hughes investigator, HHMI professor at the University of Chicago named Bruce Long. Now, you know, Bruce was at that time in the news for not so nice things, even though he was very misunderstood. And he focused on epigenetics, which was one of the chapters in my dissertation. So we really had something, and for me, I was really trying to redefine, you know, what we, you know, how we view epigenetics. So when he asked to fly out to speak to me, I figured it's a no-brainer. I have family in Chicago, take the free trip, go eat some pizza, have a good time, you know, go see a White Sox game. And I took him up on it. So I meet him at a pizza shop down in Hyde Park. And, you know, we were supposed to talk about this company, Cyogen, which was, you know, a company he started with his brother. What they did was make custom rodent models. So mouse or rat. And this wasn't my field. And what, you know, wasn't really my alley at all. But again, I wanted to just pick Bruce's brain and meet him there, you know, at the little, you know, pizza pub in the middle of the day down in Hyde Park. You know, right when I sit down and before we even get into the job interview, so to speak, he says, I've been working on this idea I want to show you. And he shows me the basic initial code for what will become Vector Builder. And we sat there for 3.5, 4 hours nerding out. And it was a meet cute. It was an intellectual meet cute. And we initially just, you know, from an intelligence standpoint, just, you know, really fell for each other and what, you know, this idea could become. He actually was teaching and he missed 3 classes he was supposed to teach, you know, as we kept talking. And we realized that, you know, we had more to talk about. And we have never looked back ever since then. So again, I'm still working for Cyogen at the time. After I joined Bruce a few months later, I tell him I want to go move out and open an office in Europe. You know, I actually go and was looking at the UK, Germany. I thought I spoke good German, so I went to Germany. It's in the middle of the country or the continent. So I ended up going out there, and we're building Cyogen. But at the time, in the back of our minds, we're like, you know, fostering our idea of vector builder and what these vectors could be. And at the time, the vector builder idea was kind of building the vectors for Cyogen. So, you know, over time, you know, as many companies do, you kind of have a competition and you have to split when one gets, you know, too big. And that's what Vector Builder was doing. Vector Builder was clearly on a trajectory higher than the mouse research could ever do. And when we hit 2020 and the pandemic, things really took off. So those of you, I mean, not many people know this. We don't really brag about it as much as we probably should. But, you know, Bruce and I we were the first people to pseudotype coronavirus spike protein in 2020. You know, basically making it safe for BSL-2 labs to study across the world. Now, you could imagine the shipping woes I encountered with customs shipping to, you know, close to 75 countries at that time. And when you declare on the customs, we're shipping coronavirus. So I was constantly on the phone trying to explain to them what a pseudotype virus meant and how we stripped everything that is actually pathogenic and it's completely safe. But it really was one of the big, you know, achievements of the company so far. And we're happy that, you know, although, you know, we're not really allowed to, you know, we're not in the news for it, because again, we are quite humble, despite how I might come off right now. We actually helped make the Johnson & Johnson or Janssen vaccine, which obviously wasn't the most popular of vaccines, but it was the only one that was one and done, which is something that we brought up that was very important for homeless people who you don't have the ability to schedule 2 shots for and don't have a phone. So I'm really happy that, you know, as of this year, Vector Builder achieved what's called unicorn status. So I don't expect a lot of you to know what this means, but unicorn status is a term that's used in the venture capital industry to refer to a private startup that has received a valuation by a third party of over $1 billion. So as of a few months ago, we received our unicorn status, and we're on Crunchbase. And this is after raising about $80 million in funding over the past 6 years. And it really is a remarkable achievement, to be a unicorn company while also caring about the researcher still. So postdocs, grad students, what do we do? What is he talking about? He's just bragging. He's just, trying to do something. So I'm going to talk about what your options are now. And before deciding that, you have to make a big decision, so to speak. And this is what I was told, at least when I was back there. Do you want to stay in academia with what I like to call the Jedi or the light side? Or do you want to come and join me in the Sith on the dark side of research? And I have always, again, being a contrarian, and I'm a huge Sith supporter. So this was a no-brainer for me. I'm not here to follow, what was done before me. I want to branch out. If you're zigging, I'm going to zag. So, that's one thing you got to decide first. The next thing is you got to ask yourself, what's your goal? What do you want to get out of, your future and your career? For me, I knew that after sitting in the lab for over a decade, that I wanted to travel the world. And like I alluded to before we started here, I said that, I think UGA was, I won a travel award to present my research in Yokohama, Japan at a conference. And ever since that trip, which was maybe 15 or 14 years ago, I've had the travel bug. I really wanted to just immerse myself in as many cultures as possible. Hence, me moving to Europe, and I've been living out here for 7 years now for the most part. I spend a lot of time, I spend a few months a year in the US or in Asia, but I really enjoy living out here in Europe, especially coming from Chicago. There's a lot less guns. It's a little bit safer out here. Since then, I've now opened offices in the US, in Germany, and the UK. And now the next step is to actually go down to Australia early next year to open up our office in Melbourne, as well as in Seoul, Israel. Might have to wait for, let's die down a little bit there before we go back to Israel, Singapore, and Buenos Aires. I actually built quite a large team now out of Buenos Aires down in Argentina. And my other goal was to pay off my student loans. So this is something that, again, is not really seen throughout the world, but it's something that I know that the current administration is trying to address. So hopefully, it's not going to be a problem for a lot of you younger people. The other thing to do is know your options. Know what you have, know what you could choose from, with a PhD in life sciences. You could go to academia, you could become a postdoc, you could be a research scientist, you could be a PI or group leader. That's becoming harder and harder to do. But, that is, what, when I signed up, that was what I expected to do. That was the normal track, so to speak. But you could also be a bench scientist at a big pharma company or biotech company. You could work for Merck, Novartis, Roche, Amgen, Pfizer, and go have a really nice, steady job, steady paycheck, good pension, and sit at the bench doing repetitive work, not really thinking outside the box, and just having a good quality of life. It's another very option, and it's very sensible. You could also go into industry, but do a more non-bench approach. This is more sales or marketing or product development, get at the business side of selling scientific, resources and products and services. You could become a scientific writer. Some people who love to read and write, this could be right up your alley. You go into the financial sector. This is actually where I was recruited to go heavily after graduating with the computer science and the genetics and then the MBA. I was looking to go to a hedge fund. I was, for any of you that do want to go this route, I will warn you that hedge funds tend to have anywhere between 7 and 10 rounds of interviews. And after getting through, I think 5 or 6 of them, we ended up parting ways because they wanted me to sit behind a bunch of computers and develop algorithms and be a quant person. And I understand that in my education dictated that, but I was a social butterfly. I wanted to travel the world. I wanted to go schmooze the millionaires and the billionaires and have the Amex Black Card and do whatever I want. But they told me that job was only reserved for actors and models and influencers and so forth. And I, begged, I didn't see eye to eye with them because I don't see why somebody who's a scientist could do the same thing. We shouldn't be pigeonholed. And finally, you could become an entrepreneur. You could go that route. And, the sky's the limit now with what you could do with, new technologies and new ideas. And if you're somebody who really has the gumption for that, but I do warn you, that is probably the hardest one out of all these pathways, is to go be an entrepreneur. It's not for everybody. You really need a certain type of personality. So we're getting close to the end here, and I just want to really kind of mention two, if you do choose industry, two things to really consider when you're looking, for where to go into and what company to pick. And it's really, you got to pick the right company. You got to believe in its technologies and future growth. Look at a company like Vector Builder. We're going to make, cloning is going to be obsolete. And what do? We do cloning. So we have nowhere to go but up. And as more and more, right now, our biggest competitor, I know I said it was GenScript, but our biggest competitor is you all sitting there. It's the researchers that still clone themselves. And I know 10 years from now, no one's going to be cloning. The amount of time and cost it takes to, to clone yourself, it's not going to be worth it. So, picking something where technology is nowhere to go but you also got to believe in upper management. You got to believe in their vision and their leadership, because that's who you're attaching yourself to, and all the people that we've hired this year. And the thing I highlighted here is you got to fit in with the company culture, especially in these days and age. You gotta enjoy your colleagues. You gotta enjoy the atmosphere, and really, wake up in the morning just enjoying to go to work. I don't wake up every morning enjoying to go to work, but I did today. And, I really hope that you guys could find, that place where you do really enjoy what you're doing. But the company culture will dictate this. And, you got to get along with your, do you want to be micromanaged? Do you want to, have full autonomy? Do you want to go into an office every day? Do you want to have the flexibility to work remote? Do you want to have to work a 9 to 5? Or are you going to, want more flexibility in your hours? These are all questions you got to ask. And I like to think that I've built a culture where I'm very proud to say that, I think I have employees in about 30 countries. And when they visit each other's respective countries, they crash on each other's couches. They stay in each other's guest room. And that's something that I really pride myself on, to keep that family type of atmosphere as we continue to expand and become one of the big, major players in the gene delivery industry. So I will leave you, because I think we're about getting close to the time, with something, and that is not to panic, especially when you're in an interview, because we as hiring managers could smell it on it. Just relax, and don't panic about having to make a decision right now or this. I would say one mistake I made is going straight from the PhD. I think I took about 10 days off and I started a position. Don't do that. Take some time for yourself. Go travel, go spend time with family, or your significant other. Just don't rush into things. You have your whole life ahead of you, and really take the time to consider some of the points I talked about today, and make the right decision for your future. And no, we're always hiring. So if you need a home, come to us, reach out to us here at Vector Builder. We could find a home for you, maybe. So I want to reward you all for tuning in here today. And that's to talk about, so, those of you who don't know, and I'm going to give a shout out here to Calvin Yee's lab, because I believe Calvin Yee's lab is the only lab that orders from us. And I hope that's going to change after you guys speak with Connie, who I'll introduce on the next slide. But we do have one of the best graphical user interfaces, or GUIs, out there. Our design platform has almost all of the functionality of a program like SnapGene, but it's all online and it's free to use. But we're not okay with having most of the features of the premier design tool like Snapgene. We want to be better. So we're actually about to launch a downloadable software desktop version called Vector B. All right, Vector B is going to hopefully be the next big, design software that exists. And not only that, as of now, and don't quote me on this for, 2 years from now, we are going to offer it free of charge to researchers all around the world. We're just finishing up the alpha testing internally on our Vector B software, and I want to invite each and every one of you to take part of the beta test. Play around with it. Give us your feedback. I can tell you if you suggest something really good, we'll reward you. We'll build vectors for you. We'll do whatever it takes. Or if you're interested in working with us, I can tell you one of the best things is help troubleshoot this right now. So if you're interested in becoming a beta tester for Vector B, please get in contact with Connie, who's going to be there, and or send me your email address. Contact our company. Say I'm a UGA researcher. I want to be a beta tester, and we'll get you on the list, and we'll send it to you probably in the upcoming weeks. So I really am sorry that I'm not there today. I'm here in Germany, and I really hope to visit the campus again soon. I do miss Athens quite a lot. It was just a very, very one of the best times of my life, and I've been all over the world, and I still reminisce about the times I had in Athens. So while I wasn't there, I'm able to send somebody who's even better than me. And that's Dr. Constance Rich, or Connie, who is there in front of you. Now she got her. She's a Georgia native, so she's back home this week. She did do her undergrad at Georgia Tech, which she apologizes for. But then she went on to go do her PhD at the University of Cambridge, which for any researcher, if you have a chance to go to the UK or you're visiting London, take the time and do a day trip to Cambridge. I mean, it really is like going into the wizarding world of Harry Potter, and, go have a pint at the Eagle. You could go look at some of the crazy things with the companies I've started. I've gone there every time I start a new company, and I put something in the ceiling or try to, instill my mark on what's, one of the notorious pubs in all the world where Watson and Crick, founded this, the structure of DNA. Connie then went on to become a lecturer at the University of Florida. She says double sorry, but Florida hasn't really bothered us, I guess, in recent years for UGA football, right? I mean, I was there back in the Matt Stafford, no Sean Marino, AJ Green days, and we could never get over that hump to beat Alabama. But now you guys are able to enjoy, all the rewards of a successful team. So Connie's now a scientific product manager at Vector Builder, and she started with us earlier this year. And it's really a pleasure to have her, and I think she's going to be better to talk to than even myself, and just really all around a great human being. So really, I encourage you, please reach out to Connie. She could come stop by your labs later, give you demos. I don't think we're gonna have enough time for a demo here, but I did promise I wouldn't plug my company too much, which I hope I didn't. But at the same time, I did definitely want to. And then finally, I just want to introduce that if you do reach out to our company and you don't go through Connie, you'll be connected with actually someone who's been with us even a few years and longer is Jessie Toomey. She's our senior territory manager. She got her, she's, a bioengineer, really, really sharp person, huge. She's a yoga instructor. So she's really easy to talk to and just a great human being as well. So really just, if you reach out and get a hold of Jessie or Connie, you're in great hands. So, with that, I just want to thank you all. We're not going to have time for the demo, and just, I'm open to any questions we have before Connie takes it over, and she's going to talk to all of you. And I really hope that I'm going to see many more orders from the UGA genetics department with Vector Builder, coming after this. So thank you. And I think I'm right on time.
All right. Thank you so much for speaking to us. I think we're running out of time, so we have time for maybe one question if someone does have a question.
I got one.
Okay.
What are the first steps to start a company?
Oh.
Let me stop sharing this. Hold on. Let me get my face on there. Okay, I want to see all you. The first steps to starting a company. Well, the easiest thing, get funding. But honestly, it all starts with an idea. It's finding an idea that doesn't exist. And I remember I went through a phase where I was coming up with new ideas, and every single one of them, this was around when I was at UGA even too. Everything was in like 2008 or 2009. There was an idea, go do your due diligence on searching if the idea exists. But if it is a novel idea, that's how companies all start, with a novel idea. And then it's about finding resources. Funding obviously is their main resource, but it's also people, professors, at different institutions that could help, you with connections and guide you to maybe the VCs to get the funding to help, fine tune your ideas. But, realistically, a company all starts, at least a good company all starts with an idea. And that idea could be a build on of an existing company. It doesn't have to be completely new, but it's got to fill a need that isn't being filled right now.
All right. Thanks so much. We got
another question. We can get another one there. Come on.
I was just wondering, so I think you said that you had 2 failed companies. What were your biggest takeaways from those failures? Or like what the most?
Yeah, no, that's a great question. I think I was very ambitious. So one of them I could tell you about was called Academic Consultants of America. What I did is I said to myself, like, after going through the journey I went through, I said, I'm gonna help people try to like decide, where to go to grad school. That was very challenging for me. When I went to UGA, Rich Meager's lab was not on my radar. I had no desire to study plants. And I ended up in his lab, and I'm just so happy that I did. So, trying to understand personalities and things like that. But then I realized that trying to help, potential grad students, they don't have money. They can't pay you. And like, you got to think about the full person. You got to sometimes think about how are you going to, generate revenues. But really, like, that was a good idea of realizing that even though I thought I had a good idea, I didn't think it all the way through. And this, along with, coming up with ideas that maybe are, could infringe on other IP that exists in other countries, or that just, you don't have the resources to reach the amount of people that you want to reach. Because that takes investment. That takes strategic planning. So, I could tell you, if you're going to go be an entrepreneur, you will likely fail, just like we do, every other day in the lab. But don't let it hold you down, because if you stay persistent, you will break through. Either that, or like I said, if you're an outside thinker like that, come to Vector Building. We'll have a place for you.
All right, thank you so much. I think we are out of time and need to leave the next class. But thank you for speaking with us, and I'm sure if anyone needs the Vector, they will come to you.
Yeah. Thank you so much. Yeah, please talk to Connie. Connie is awesome, like really, truly awesome. I can't say it enough. So thank you all.